We're hiding with a true highlight, a presentation by Dr. Teicher. And just some bio on Dr. Teicher. He is an associate professor of psychiatry at Harvard Medical School. He received his PhD in psychology from Johns Hopkins and his MD from Yale. He's a member of several editorial boards, including the Journal of Child Psychology and Psychiatry. He has published over 200 articles in top-notch journals: Nature Reviews, Neuroscience, Biological Psychiatry, JAMA Psychiatry. He has been awarded over 19 US patents, and he is also the recipient of numerous awards, including the Memorial Award for Outstanding Scientific Achievement from the International Society for Traumatic Stress Studies. His research, as you all know, focused on the neurobiological impact of trauma, particularly in childhood, and how these experiences affect mental health across the lifespan. Specifically, his lab looks to identify sensitive periods where the brain is most vulnerable to early life stress and associated mental health risk. He also investigates brain network changes related to resilience and recovery. And his lab develops tools to diagnose, assess, and evaluate treatments for psychiatric disorders. So his talk today is titled Childhood Maltreatment, Windows of Vulnerability and a Novel Understanding of Resilience and Recovery. So with that, a very warm welcome to you, Dr. Teicher.
Oh, thank you so much, Stephanie.
You're welcome.
My pleasure. Okay. So let me begin by giving a definition of childhood maltreatment. This is from the World Health Organization. It's a nice broad definition. All forms of physical and or emotional ill treatment, sexual abuse, neglect or negligent treatment, or commercial or other exploitation, resulting in actual or potential harm to the child's health, survival, development, or dignity in the context of a relationship of responsibility, trust, or power. So maltreatment is highly prevalent. About 15% of adults without psychiatric histories have self-reported moderate to severe maltreatment. About half of the adults with schizophrenia, bipolar, or major depression report moderate to severe maltreatment, and 3/4 of adults with persistent depression report maltreatment history. So we see it a lot in our clients. That's not here. So the biggest question is, how does stress affect the brain? And when I started doing this research, I think current thinking, Doug Bremner had just published a book on, does stress damage the brain? Basic thinking was that stress was bad for the brain, and particularly bad for the developing brain, and that it was, through high levels of cortisol, affecting neuronal viability and damaging the brain in a variety of non-specific ways. And after I was working on this for a while, it sort of dawned on me that this really didn't make sense. That, through all the eons of evolution, wouldn't a mammalian brain that was resilient or resistant to the effects of early stress, be selected? And so the logical alternative is that exposure to early stress generates molecular and neurobiological effects that alter neurodevelopment in a potentially adaptive way that prepares the brain to survive childhood and to reproduce in what it's predicting to be an experience of potentially malevolent world. So we think about these brain changes not as non-specific damage, but as potential phenotypic adaptations that in the short term might be beneficial in terms of survival and reproductive success, but in the long term may have many maladaptive components. So the first principle is that maltreatment does not in general result in non-specific damage, rather it produces specific phenotypic modifications that may foster survival and reproductive success, but may also be, particularly at later ages and other settings, highly maladaptive. So what might be some beneficial phenotypic adaptation? So if you're living in a situation where you may be, abused, and there may be particular severe threats, that enhanced ability to detect and respond to threats may be a beneficial adaptation. So here is an outline of neural processing of threats and response based on a number of different models that we've kind of combined. The important point from this is that there's two pathways. There's both a low road and a high road. Information from most senses comes into the thalamus, and there's a pretty direct connection between the thalamus and the amygdala. And this can work in just a short number of milliseconds to generate a response. And that this can happen at the level of the hypothalamic pituitary adrenal axis. You can also have through the periaqueductal gray and the bed nucleus of the stria terminalis and the nucleus accumbens, defensive reactions and defensive actions. There's also the high road, in which information goes into sensory cortex and then comes into your perceptual awareness. This gets processed. It perhaps comes into your full conscious awareness when it gets into the level of the ventral posterior lateral cortex, and it has a bunch of regions that can then modify the amygdala response. It can bring in contextual information. It can also exert top-down regulatory control, or it can also enhance amygdala response. So here's a complicated circuit that's very important for survival. And the question is, how is this modified by childhood maltreatment? Oops. So we'll focus on some of the key regions. We'll focus on the amygdala, the hippocampus, the thalamus, portion of the ventral posterior lateral, the ventral prefrontal cortex, other portions of the medial prefrontal cortex, and sensory cortex in terms of how we process this information. So looking first at the amygdala, you see it here, almond-shaped nuclei. The amygdala is a key limbic structure that's critically involved in encoding of implicit emotional memories and detecting and responding to salient stimuli such as facial expressions and potential threats. And structural functional abnormalities in the amygdala have been reported in a vast array of psychiatric disorders, almost everything, PTSD, phobias, unipolar and bipolar depression, drug addiction, autism, a lot of personality disorders, schizophrenia. We know from animal models that exposure to stress leads to persistent neuronal hypertrophy and symptoms of anxiety. So the pyramidal cells have an expansion of their dendritic arborization. These are very persistent. They don't reverse with time. Even when the prefrontal cortex comes in to have more of a regulatory control over the amygdala, these abnormalities don't reverse. These are enduring changes that are produced by stress, particularly early stress. If we put individuals in the scanner and expose them to threats, you can see enhanced activity of the amygdala, and particularly interestingly on the right side, and that this is influenced by childhood maltreatment. The higher your childhood trauma questionnaire scores, the more on average is your amygdala response heightened by this exposure when you're viewing, say, threat versus neutral stimuli in the scanner. So a big question is, what particular types of exposure and at what ages affect how your amygdala is going to process this information, and what kind of experiences at what ages might affect the actual morphological development of the amygdala. And so working on the idea that there are sensitive periods, that there are particular stages during development often when brain regions are going through rapid change, that they're particularly vulnerable or modifiable by the effects of stress, and that there are these windows in which these brain regions are plastic. Actually, what we understand now in a much clearer way is that there's, over the course of development, there's excitatory neurotransmission tends to develop more rapidly than inhibitory neurotransmission. And at a certain point, they come into balance. And when the inhibitory and excitatory processes come into balance, it opens up a window of plasticity in which brain regions may be very affected by their environment, and that we know that the brain is much more plastic during development than later in life. But interestingly, it's not because the brain stops being plastic. It's because there are molecular signals that come in that actually impede the plasticity. So there are brakes that are put on to prevent the plasticity, but we have periods of time in which the brain in specific regions are particularly, plastic. So we're trying to identify, these sensitive periods and to understand, what's happening. And what's turning out in our research, we'll talk about a bit, there may be multiple periods of plasticity for a brain region, not simply one. So a lot of this work in the beginning did with Caroline Lyons-Ruth, who is an attachment researcher at Harvard, and she has recruited a sample of individuals that she studied for attachment using the strange intruder paradigm, and she's continued to follow these individuals for over 30 years, and we got a small amount of funding to bring these in and to scan them and to look at brain in relationship to early attachment. And so we brought in a small sample, compared them to healthy young adults where there's no question about exposure to adversity. And we looked at the volume of the right amygdala and looked at the volume of the left amygdala. And we found that overall, that in this group that we brought in, the amygdalas were slightly larger on both the left and right side. On the right side, there was a clear relationship between their degree of exposure to adversity and the volume of the amygdala. More on average, the exposure to adversity, the greater on average was the amygdala volume. On the left side, there wasn't a significant association. So it seemed like the right was affected by adversity, not the left, even though both were enlarged. We did a sensitive period analysis and found that it was particularly exposures at 10 and 11 years of age that were associated with a maximal effect on the volume of the amygdala. But the question is, what's going on with the left? And when we looked at the attachment data that was collected 30 years earlier, we found that the left side correlated with attachment behavior at 18 months of age. And the more disorganized the attachment, the more disrupted the communication, the greater on average was the volume of the left amygdala 30 years later, which suggests that that this is being responding to this very early experience. Now, it's interesting if you think about the left side and the right side. We know that the amygdala is playing a critical role in detecting and responding to threats. And if you think about it from a developmental perspective, there are two different classes of developmental threats. The most dangerous would be rejection or neglect, of the early infant. There's also other threats that may come in the form of abuse. And so we may have two somewhat different threat detection systems. You might have a left-sided system that's specialized in some way for detecting aspects of rejection that may be in childhood detecting disruptions in attachment bond, maybe later in life detecting alterations in our adult attachments, and the right side detecting other types of threats. This makes sense also if we think about differential function of left versus right hemisphere. There's a number of different models that talk about hemispheric laterality. The valence model particularly talks about the left hemisphere being specialized primarily for the expression and emotion of positive emotions, and the right hemisphere being specialized for the expression of negative emotions. Another way that people look at it is in terms of approach avoidance. The left hemisphere seems to be specialized in approach-related emotions, the right with avoidance or withdrawal-related emotions. It's generally positive emotions that are associated with approach, negative emotions that are associated with withdrawal, with one major exception, which is anger, which is also associated with approach. And so it makes sense from the standpoint that if you're detecting a relationship threat, that approach may be the appropriate response, whereas you're detecting other sorts of danger, that withdrawal may be the more appropriate response. So interesting to think that we might have these two somewhat different threat detection systems. And that's talking about the amygdala from a morphometric standpoint, its volume. What about in terms of the amygdala's response? Lots of studies have looked at the amygdala response to threatening faces, and you can see abnormalities in a whole host of disorders, particularly anxiety disorders, major depression, PTSD, drug abuse. It's also turned out that some studies have pointed out that the amygdala is having a hyper-reactive response, that it's responding much more strongly in some individuals with psychopathology than in healthy controls. And that's particularly true in the case of individuals with anxiety disorders or PTSD. There are also instances in which the amygdala is having a blunted response and is not showing a strong response to threat. And that seems to emerge in individuals with disinhibited behavior, taking risk-taking, inappropriate social behaviors. They may not be detecting threats as well. And so the question is, are there situations, types and timing of maltreatment that lead to a hyperactive amygdala response, and are those that are associated with a hyperactive amygdala response? So we had a reasonably large number of participants, and we looked at and assessed their type and timing of exposure to maltreatment. And we did this using an instrument that we call the Maltreatment and Abuse Chronology of Exposure Scale, which looks at 10 types of childhood maltreatment. And we get an idea of the severity of exposure to each of these 10 types of maltreatment across each year of childhood. So we wind up with a tremendous amount of data. Not all of these actually occur. You don't have to worry about being exposed to pure emotional abuse at 1 year of age. But that many of these, about 140, wind up as potential predictors in a analysis. And you can see that the different types of maltreatment have their own unique time course. We're looking here at male versus female differences. And you can see that certain types of maltreatment, like emotional neglect and physical neglect, are fairly flat across development. Parents don't change very much in terms of either becoming neglectful or non-neglectful at different points. You can see that other things like parental physical maltreatment peaks relatively early in childhood. Aspects like parental verbal abuse or parental nonverbal emotional abuse tend to increase into the teenage years. You can see that early sexual abuse, that there's no specific male-female predominance, but beginning around age 7 or 8, becomes much more prevalent in females. You can see that pure physical bullying becomes more severe in males. And so we can get a good idea of how these things change developmentally. As an aside, the question is, how distressing is it to provide detailed self-report information on exposure to childhood maltreatment? We have the situation, we started doing this, and Al Khan has been working largely on this, where our IRB required us, when we started collecting this information, to provide a phone number with 24/7 coverage, just in case individuals had a negative response to providing detailed information on their exposure to adversity, particularly, questions about exposure to different types of sexual abuse. And there's been situations where I've heard from others that their IRBs have not let them use the MACE. These are generally at colleges that don't have a medical school. We've now had over 3,000 individuals complete the MACE. We've never received a phone call in terms of distress. We have received a lot of correspondence indicating how helpful they found the scale, but nobody's called in a state of distress. So we wanted to test the hypothesis that completing the MACE would be no more distressing than doing something like a timed scholastic test, which does not require IRB approval. And so we collected data using prolific We looked at their distress using the abbreviated POMS, and that will give us information about their feelings of tension, anger, fatigue, depression, esteem-related affect, vigor, and confusion at the moment when they're experiencing it. And we had about almost 500 participants, mean age of 42 years. And here is their overall total mood disturbance score. This is right before they did either the POMS or the academic test, and this is afterwards. And you can see that the MACE increases slightly. Their mood gets much worse after doing the academic test. Now that's for all the subjects in general, and we see that it's much more stressful doing a timed academic test on average than doing a self-report measurement on your adversity. What about individuals who actually have high levels of adversity? Here's a distribution of scores ranging from no exposure to adversity up to 10 types of adversity. We've broken into categories 0, 1, and 2, and then 3 or 4, and 5 to 10. And here's your distress doing after your academic test is high, and is high in all groups, about the same. In the individuals who had no exposure to early adversity, they experienced very little distress, which went up with their level of adversity. But basically, even at the highest levels of adversity, it's no more distressing than doing a timed academic test. It's useful to use this kind of assessment and evaluation, and one doesn't have to worry about precipitating all sorts of psychiatric problems by inquiring about their exposure. Zhenjun Zhu was a graduate student from China who completed his thesis in the lab, and he's been doing a lot of work on the sensitive period in terms of functional behaviors in the brain. And we did a study in which we exposed individuals to either angry or threatening faces, neutral faces, or simple geometric shapes. And we use machine learning to identify, oops, that slide keeps on flipping, type and timing of maltreatment that are the most important predictors of response. These are the 10 types of maltreatment: emotional neglect, nonverbal emotional abuse, pure physical abuse, parental physical maltreatment, physical neglect, parental verbal abuse, sexual abuse, witnessing into parental violence, and witnessing violence to siblings. And good grief. And these are the ages of exposure, and the analysis, which is random forest with conditional inference trees, is telling us what predictors are the most important in terms of how much permutating one of these predictors will decrease your goodness of fit. And we can see that particularly nonverbal emotional abuse at around age 9 is a powerful predictor. We have early pure physical maltreatment, and later pure physical maltreatment. We have later pure verbal abuse. We have witnessing into parental violence. These emerged as significant predictors. These over here are the severity of maltreatment, the duration, the number of different types of maltreatment, and the duration of maltreatment, which are not as important as some of the specific predictors. And what we found is that there were some predictors, like pure physical maltreatment at age 6, witnessing into parental violence at 7 and 9. These predictors all predicted a blunted response. These later teenage predictors predicted an enhanced response. And we see, if we look at unexposed individuals, you can look at the response to the negative face, response to the neutral face over time, that the bold response to the negative face is stronger on average than the bold response to the neutral face. In these individuals who've had just this later exposure, you can see that it's greater, that there's a much greater response to the negative face than the neutral face. And individuals who've only had these early exposures, but not these later exposures, have the opposite. They having actually a greater response to the neutral face than they're having to the negative face. They're having a blunted response to the negative face and an enhanced response to the neutral face. So we have two sensitive periods, one sensitive period producing an enhanced response to negative faces, another sensitive period producing a blunted response to negative faces and enhanced response to neutral faces. So turning to the hippocampus, again, major portion of the limbic system, critical in the formation and retrieval of explicit memories, including autobiographical memories. They've also been reported in many different psychiatric disorders: PTSD, depression, schizophrenia, bipolar disorder, drug addiction, borderline personality disorder. Interestingly, stress has, at the cellular level, the opposite effect on the hippocampus that it does on the amygdala. Uh, Instead of increasing the dendritic arborization of the pyramidal cells, it actually suppresses it, and it causes retraction of their dendritic arborization. It also has the additional effect of suppressing neurogenesis in the dentate gyrus portion of the hippocampal complex. These effects, however, are more reversible than the effects on the amygdala. And the research on the hippocampus was done in the lab by Carl Anderson and Kyoko Ohashi. And in one of our studies, we looked at this and we simply simplified the analysis to look at exposure to two different types of adversity. We combined all of those that were measures of abuse, and we combined all of those that were measures of neglect to look at this. This provided a nice, simple understanding of the hippocampus. We can also talk about this in terms of threats, which are the abusive aspects, or aspects of deprivation, which are the neglect aspects, which Katie McLaughlin has talked about as two fundamental two fundamental aspects of maltreatment. And what we see that completely different story in males and females, that hippocampal volume in males is affected by neglect, particularly during the first 7 years. It is not particularly affected at all by abuse. In females, it's not affected by neglect. It's affected by abuse, and particularly around 10 or 11 and 15 or 16 years of age, that we get effects of abuse on the female hippocampus. In terms of the functional response, we found the same kind of story, that there are early experiences that lead to a blunted response, particularly around 9 or 10 years of age. There are later experiences in the teenage years that lead to enhanced response. You can see the normal difference is pretty modest. It's much larger in those who've had this later experience, and it's blunted completely in this case in individuals who've had the early experience. So turning next to sensory cortex and thalamus and these pathways. This was essentially a series of studies that we did asking the question, does the nature of maltreatment matter? And this was done by Ji-wook Choi, visiting scientist from South Korea, and Akimi Tamada, visiting scientist from Japan. And we looked at different types of adversity, and we looked at it in two ways. We did fiber tracks to analyze white matter, and we used gray matter to look at voxel-based morphometry. I point these out because these are unbiased whole brain analysis. So we're basically taking all the data, providing it to the computer program, and asking the computer program just in an unbiased way to identify the brain regions in which we may have the biggest group difference or the biggest association with exposure to a particular type of maltreatment. And what we found in a series of studies was this, that in individuals who had high-level exposure to parental verbal abuse and no other exposure to adversity, that it specifically seemed to affect the superior temporal gyrus and the portion of the temporal gyrus around the auditory cortex in terms of morphometry. And actually, it was an increase in gray matter volume in this portion. It also affected the integrity of 3 fiber pathways. The one that was most affected is the left arcuate fasciculus. And the left arcuate fasciculus interconnects Broca and Wernicke's area as an important language pathway. And the greater the disruption in the integrity of this pathway, lower their verbal IQ and comprehension. And this is particularly assessing the auditory cortex on the left side. In contrast, if we look at the effect of witnessing domestic violence, and in this case, we recruited individuals who had at least 3 episodes of visually witnessing domestic violence between their parents. What we found was that there was alterations in the gray matter volume in the visual cortex, particularly primary visual cortex. There was a substantial reduction in gray matter volume. And there was also a significant effect on one pathway, in this case, the left inferior longitudinal fasciculus. The inferior longitudinal fasciculus connects the visual system with the limbic system and determines our memory and emotional response to things that we see. So basically, just think of the remarkable specificity of this, that the verbal abuse is affecting auditory cortex in an important language pathway. Visually witnessing domestic violence is affecting visual cortex and an important visual limbic pathway. What we found is in individuals who had experienced childhood sexual abuse, that it also affected the visual system, primarily the V2 as opposed to V1, and involved in regions involved in facial recognition, regions that seem to be involved in dream imaging. Similarly, Christine Heim in her laboratory examined women who had experienced multiple episodes of penetrative sexual abuse in childhood. And what she found was that there was specific thinning of the somatosensory cortex. And the specific thinning occurred in the portion of the somatosensory cortex that represents sensation from the clitoris and surrounding genital area. So that basically maltreatment targets the sensory systems that convey the information, and perhaps in an adaptive way by tuning down these systems so you don't experience as intense trauma as you would if these systems weren't turned down. And that may be adaptive in the short term in terms of surviving your childhood, but it also has long-term effects. What Christine is finding is that by affecting this aspect of the somatosensory cortex, you're affecting ability to experience sex, and that you may have real problems with orgasm or a whole different attitude towards sex. And this biological effect may be one of the reasons why one of the consequences, it seems, of sexual abuse is a high risk for prostitution. Maybe because these individuals have different attitudes or feelings about sex. And I think it's also associated in some of our patients with anorgasmia. This is just data confirming that the parental verbal abuse really targets the superior temporal gyrus in males in a much larger sample. Interestingly, all the sensory systems are affected, but what about effects on the thalamus, which is our primary sensory relay nuclei? It's turned out that most studies have not reported any significant effect on the thalamus. And a big question is, how many types of maltreatment and how important is age of exposure in terms of finding this effect? And we looked at this, and what we found briefly was that if you looked at it in a simple way, if you looked at it and compared maltreated to non-maltreated simply, you really didn't see much in terms of the thalamus. If you looked at whether they experienced threat or whether they experienced deprivation, you saw a slight effect. If you looked at it in terms of number of types of maltreatment, you saw a slight effect. But basically, you found very little, only perhaps a slight enlargement in thalamic volume, and particularly just on one side. So here you might see there's a slight, with two-group comparison, increase in volume on the right. If you looked at it in terms of number of types, in this case, you see a slight increase on the left and only in those who had 3 types of maltreatment. If you did deprivation or threat, you saw very little. But if we broke it down into, in this case, 4 categories: emotional neglect or deprivation, physical neglect or deprivation, physical or sexual threat, and emotional threat, and looked at it across age, we got a very different story that we found in terms of the right thalamus, that emotional threat early on affected the volume, physical threat in later years affected the volume. And interestingly, early emotional threat led to an increase in thalamic volume. The greater the exposure, the greater the increase in volume, whereas physical threats led to a decrease in volume. Similarly, on the right side, early threat, later threat, early emotional threat, increase in volume, later physical threat, a decrease in volume. So understand why studies weren't showing it is because some exposures were leading to an increase in volume and some exposures later on were leading to a decrease in volume. So just looking at it in terms of overall exposure, you may see very little. What we're able to show is that this was also specific to different portions of the thalamus. There are the different relay nuclei. There's the lateral geniculate nucleus that's relaying visual information, the medial geniculate nucleus that's relaying auditory information, the ventral posterior lateral that's relaying somatosensory information. And these are all reduced by exposure to physical sexual threat around 9, 10, 11 years of age. Whereas there's also other portions like the ventral anterior magnocellular portion, the central medial portion. These are the ones that are enhanced by exposure to emotional threat. And these are involved in movement and motor control, motor planning, and this is involved in emotional regulation and cognition. And so we're getting a complicated story where different portions of the thalamus are affected by these exposures, but only if you look at it in a way in which you have information about the type and timing of the exposure. Basically, as you all know, the brain goes through a massive degree of development. This is looking at the neuronal structure at the time of birth, and you can see this high density of interconnections that occur at 6 years of age, as there's a massive overproduction of these of synapses and dendrites and axons and in their interconnection. And then there's a pruning of these processes. And what we're basically understanding is that early exposure probably works through the overproduction process to provide these phenotypic adaptations, and that later exposure probably works through the pruning process to also provide the phenotypic adaptations. So early exposures may be associated with increases in volume. Later exposures may be associated with decreases in volume, both of which are producing phenotypic adaptations. So those with a prepubertal overproduction, enduring volume increases, peri and pubertal exposure result in volume decreases. Turning to the prefrontal cortex, there are various portions in terms of the anterior cingulate, the dorsal and the ventral medial prefrontal cortex, orbital prefrontal cortex. These are all important in emotional regulation. They're all affected by maltreatment. Just summarizing the literature, the vast majority of studies show decreases in anterior cingulate, decreases in the orbital frontal and ventral medial prefrontal cortex, decreases in the dorsolateral. In terms of the effect on function of these regions, similar story. There are later exposures around the teenage years that lead to a heightened response in terms of the anterior cingulate. There's some early exposures that reverse the situation in which you're having the greater response to neutral stimuli than response to threatening stimuli. That's true for the dorsal anterior cingulate. It's true for the ventral medial prefrontal, and it's true for the inferior frontal gyrus, pars triangularis, that that you have these early things that enhance response to neutral stimuli and later things that specifically enhance response to threat. So what do we make of these consequences and alterations in bold response to threat? Ian McCrory has an interesting theory that he sees these alterations in functional response as latent vulnerabilities, that they increase the risk for developing anxiety disorders rather than producing symptoms. And we basically find the same thing, that you can have these alterations in the brain, but in some, they may be relatively asymptomatic. and in some they may be associated with symptoms. And there's something else that will generally lead to their emergence as symptoms at a different point in development. But basically, they substantially increase your odds of having this. An increase in the negative versus neutral response will increase your odds by about 5.5-fold of developing an anxiety disorder. And if it goes in the other way, within ventral medial prefrontal cortex, it markedly diminishes your odds of, of having a response. So maltreatment exerts prominent effect on key components of the threat detection and response circuit. Early exposure can lead to a blunted response. Later childhood exposure can lead to enhanced response. And sensory systems are our primary window to the external environment are selectively modified by the types of abuse they convey. We don't have time to get into it. There are also prominent effects of maltreatment on the reward system. In general, maltreatment is associated with blunted striatal response to anticipation of reward. It's also often blunted receipt of reward, though some individuals may have an enhanced response to receipt of reward, but it does markedly affect the reward system as well as the threat detection system. It also affects integrity of the corpus callosum, which interconnects the right and left hemisphere. And it does this in a gender-specific manner. Interestingly, in females, maltreatment affects myelinization of the corpus callosum, whereas in males, it affects the integrity of the axon. But in both cases, you get decreased connectivity between your left and right hemispheres. So let's turn my attention to circuits and networks. This work was done by Kyoko. And the big question that we're trying to address is, why is it that some individuals with maltreatment have terrible psychiatric symptoms? They're really affected. It's really, had enormously severe consequences on them in their life. And other individuals with similar exposure seem to be doing much better than you would predict, given the severity of their exposure. And so the first question, are the brains of individuals with histories of maltreatment, but without psychiatric symptoms, are they unaffected? And our initial thinking was that the psychiatric and neurobiological consequences would go hand in hand, and that individuals who had been exposed to significant maltreatment and who had psychiatric alterations, that they would have big changes in their brain, whereas comparably maltreated individuals who were free of psychiatric symptoms, well, we figured their brains would look much more like the brains of individuals who weren't exposed to maltreatment. And like many of my hypotheses, that turned out to be completely wrong. These are all the abnormalities that have now been observed in individuals who have experienced maltreatment, but who don't have significant psychopathology. And we can see that these prefrontal regions, the hippocampus, the functional activity in the amygdala, and the integrity of the corpus callosum, these other pathways, they've all been reported to be abnormal in individuals with maltreatment, but without psychopathology. So maltreatment is associated with marked alterations in brain structure and function and connectivity, even in individuals with no history of psychiatric symptoms. This suggests that psychiatrically resilient maltreated individuals are not neurobiologically unaffected. Rather, they're effectively compensating through other mechanisms. And the critical question then is, how do they manage neurobiologically to maintain mental health despite a host of abnormalities in stress-susceptible structures? So we studied this by taking our sample of individuals who we've neuroimaged and gotten good MACE data on, and divided them into 3 groups. And we had an unexposed group that didn't have very much exposure to any of the 10 types of maltreatment. And we had 2 groups of individuals who were maltreated and whose exposure to the 10 types of maltreatment were highly comparable. But one of these groups was highly symptomatic, and the other group was no more symptomatic than our healthy controls in terms of depression, anxiety, somatization, anger, hostility, risk for polysubstance use, or amount of alcohol that they were consuming. So we had a control group, we had an asymptomatic maltreated group and a symptomatic maltreated group, where these 2 groups had very comparable exposure to maltreatment. And we looked at network architecture of their brain, and there's a lot of measures that you can take. You look at interconnections between all the brain regions. We're using diffusion tensor imaging to see the wiring diagram of the brain, basically how much each region was connected to each other regions. And you can look at parameters such as the global efficiency of the network, the small worldedness of the network, and the vulnerability of the network. And you can see that they had reduced global efficiency, enhanced small worldedness, and enhanced vulnerability, but it didn't matter if they were asymptomatic or symptomatic. They had the same alterations in network architecture. So we hypothesized that resilience occurs in maltreated individuals with vulnerable networks and abnormally functioning nodes, and the results may be a result of diminishing the impact of potentially problematic nodes. So here's basically the way networks are set up is you have in a small worldedness network, you have communities that are interconnected, you have high integration within communities and less integration, so more separation between these communities. And you may have a node within a community that's highly problematic, and this node is projecting information all throughout the network. If you had a problematic node and it was somewhat less connected, then maybe the network can then compensate for a misfunctioning node if this node is having less influence than in this situation where the node is having a lot of influence. So we looked at this, and we looked at a measure of nodal efficiency. It measures the central importance of a node and indicates the ability of a node to spread information to other nodes in the network. And we figured that the right amygdala is often hyperreactive in individuals who've had maltreatment. Maybe in the resilient individuals, this node is less connected than in the susceptible or in the controls. And so we looked at nodal efficiency. Here are the symptomatic, the susceptible individuals. Here are the asymptomatic resilient individuals and the controls. And we can see that there's reduced nodal efficiency in the right amygdala. And we also then identified 9 other brain regions that differed between these groups. And that in all cases, didn't have to be, they could have been increased nodal efficiency or decreased nodal efficiency. But in all of these cases, we had a situation in which these 9 nodes were decreased in nodal efficiency in the resilient individuals. May seem confusing that nodal efficiency is associated with resilience has a normal inclination would be to believe that resilience should stem from enhanced connectivity. But when it comes to the brain, bigger may not be better, less may be more. Postnatal brain development goes through both an overproduction and a pruning phase with marked increase in efficiency and performance, resulting in the strategic pruning of connections. It's network design. It's not beneficial for nodes to have as many connections as possible. They need to have an optimal number and configurations of connections. And in the sparse network of the maltreated individuals with fewer frontal hubs, it may be optimal for certain nodes to have reduced connectivity or nodal efficiency. What about these resilient nodes? I won't have time to get into all 9 of them. The other node that's particularly interesting besides the right amygdala is the left inferior pars triangularis. And we see that if you look at individuals' exposure to maltreatment, if they have high nodal efficiency in this region, they develop severe symptoms of depression with maltreatment. If they have low nodal efficiency, they seem to be resilient to the effects of maltreatment on levels of depression and anxiety. The left part triangularis contains a portion of Broca's area, and it seems to be responsible in large part to the internal voice that we have in our head, and that maltreated individuals also often have this self-castigating, intrusive voice, and that this may be the product of the left pars triangularis. And if we reduce its connectivity, particularly reducing its connectivity to portions of the limbic system, may make individuals more resilient. The other thing about resilience is that there may be 2 aspects. There are individuals who have what we call a minimal impact resilience pattern. They can be exposed to maltreatment and develop very little in the way of symptoms. There can be other individuals who become initially symptomatic, but then recover. And what we found was that in the minimal impact group, that they had reduced nodal efficiency in basically almost all of these nodes. whereas the recovered group only had reduced nodal efficiency in the right amygdala and the right subcallosal gyrus. And so it may be the case that these individuals had pre-existing differences that made them fully resistant and not experience worsening symptoms with exposure. These individuals may have been very much like the susceptible individuals, but over the course of time, changes took place in these two nodes that allowed them to recover. And this is particularly interesting, as it may be the case that to treat individuals and to facilitate recovery, all we may need to do is to change the connectivity of these two brain regions. And so that gives us a different idea of treatment. We don't have to reverse the whole panoply of abnormalities that have occurred, but we may only need to target a couple of changes that could lead to recovery. So effective treatments for maltreated individuals with psychiatric symptoms, they work by reducing the nodal efficiency of selected nodes, particularly the right amygdala and the right subcallosal gyrus. The last part I want to talk about is the idea of ecophenotypes. We know for highly prevalent disorders like major depression, anxiety, PTSD, substance abuse, there's a substantial subset of individuals with maltreatment histories, early life stress, and a substantial subgroup that don't. About 50% of these individuals will have maltreatment history, about half won't have a maltreatment history. And we're thinking about the maltreated subtype as an ecophenotype, and that we've hypothesized that the maltreated and non-maltreated individuals with the same primary DSM diagnosis are clinically, neurobiologically, and genetically distinct. The ones with the maltreated subtype have an earlier onset, on average more severe course, more comorbidities, greater symptom severity, and poor response to treatment. This difference can be quite dramatic. In a study by Williams, where they reanalyzed, I think, the STAR-D study, they looked at response to SSRIs in those with new onset depression. And if you look at those who had no exposure to adversity, you had 84% of those individuals remitted. If you look at those who had maltreatment, particularly between 4 and 7 years of age, you only had about 16% remit. So the differences can be astonishingly great. The ecophenotype also has autoimmune, metabolic, cardiovascular, inflammatory, and obesity-related problems that you don't see in the non-maltreated subtype. And we just recently published a paper in psychiatric research showing that big risk factor for obesity in inpatients was this history of childhood maltreatment. And it eclipsed the impact of medications. That's really maltreatment that drives so much of the obesity. You'll also see hippocampal and amygdala differences in the maltreated individuals with these different disorders, but not in the non-maltreated individuals. There's about 9 different psychiatric disorders that are associated with reduced hippocampal volume. All of these are increased in frequency by childhood maltreatment, and it's childhood maltreatment that drives the reduction in hippocampal volume. So what we've been thinking about is that in the maltreated individuals, you'll often see morphological abnormalities and stress-related substructures. You will not see them in non-maltreated individuals. They tend probably to have more neurochemical or functional abnormalities or alterations in different structures that aren't necessarily the stress-susceptible structures. So maltreated and non-maltreated individuals with the same primary DSM diagnosis appear to be clinically and neurobiologically distinct. This observation has far-reaching ramifications for diagnosis and treatment and research. I think really need to emphasize that it's really, I think, important in clinical trials to be able to collect data on histories of maltreatment so you can talk about whether this treatment is as effective or not in maltreated individuals. Most treatments so far to be less effective in maltreated individuals, but there's one paper that provides data that ketamine may actually be more effective in refractory depression if they have a maltreatment history than if they don't have a maltreatment history. So that's it. And here's current staff and colleagues who've really helped in doing all this work. Thank you.
Fantastic. Thank you so much. We have a couple minutes for questions. Just shout it out.
That was remarkable. I just put something in the chat because I wasn't sure if I was going to have time to say thank you to you. But hi, Nikki Bush. Your work is really influential and super, super powerful. You just gave an astounding overview of it. I don't think there's time for the 1,000 questions I have, but I think one of the things that so many folks on this call will be wondering are what's next on what you want to unpack and dig into. There's a lot of postdocs, a lot of junior faculty on this call. Not everyone does work in this space, but it would be really interesting to hear what you're chewing on these days, because you've answered so many questions. Obviously, it needs to be replicated larger. We need to better understand as technology advances and all of those things. There's a lot of work left to be done in this area, but what are you thinking about these days?
That's a great question. So some of the work we're doing is focusing more on the early exposures, and looking at the effects on attachment and how that can be lead to alterations in both the infant brain, lead to intergenerational transmission. So working on that. What I'm really interested in, trying to get funding to do this, is to see if we can prevent or preempt the effects of maltreatment. So we designed a study looking at children who've been exposed to maltreatment who haven't yet developed much in the way of symptoms. And what we found is that there's often a large gap, that there's on average about a 9-year gap from when individuals first experience, say, first episodes of sexual abuse and go on to develop major depression or PTSD. And the question is, are there some interventions that we can do early on that would really, preempt their development of psychiatric illness? And I think as a field, we need much more in the way of prevention. Treatment's hard if we can really prevent it. And can we develop, some biomarkers that may say, okay, you've had some brain changes, you're at risk, or you had some alterations in your autonomic nervous system or some other symptoms that may suggest you're at risk. And maybe there are some either psychotherapies or mild somatic treatments that may be effective. We're also doing some very interesting work with transcranial photobiomodulation. We're having some nice effects with that, and particularly looking at differences in left and right hemisphere, with the idea that these maltreated individuals have a smaller or thinner corpus callosum and more in the way of left-right hemisphere differences, and that the psychiatric symptomatology may in part be the result of a hemispheric imbalance, and that targeting the hemisphere with a more positive emotional valence seems to have a rather dramatic positive effect. We've actually been using this now in a study of individuals with opioid use disorder. And it's a very, very severe sample. We have all of these individuals are abusing fentanyl. In terms of symptoms, 6 or more is considered severe. Our least symptomatic person has a 9. And using transcranial photobiomodulation targeting towards the positive hemisphere. It's a randomized double-blind controlled trial, but we're having a significant number of individuals markedly reduce their use of fentanyl, and some of them even completely coming off fentanyl with no other treatment.
That's incredible. Thank you for sharing that. I have to run. I'm so grateful for your work all these decades. Really appreciate it.
Thank you. Thank you.
And now we're really out of time, but maybe one more question, but also can stay a little extra. Well, that's my chance to ask my question. So we talked a lot about the early developmental years, right? The early childhood. What about in midlife? Like 40, 50, is there what's the plasticity? Is this, of course, earlier is better to intervene, but are there still options available for those who are in midlife and beyond?
A great question. Well, I think it's a tough question. I think that I think we run into a wall in psychiatry in which we can make adjustments, we can make changes, but it's hard. And I think that's the promise of the psychedelics.
Mhm.
It's the promise of things like ketamine, that they enhance levels of BDNF. I think they make the brain more plastic. And so I think we really need, going forward in the future, to be able to combine our therapeutic manipulations with things that may make the brain more plastic, so that these changes that we're inducing can really stick. But I think, I think treatments like DBT are, are quite effective and can have transformative effects on people. So even without it, but I think if we can marshal, these treatments and combine it with something that will enhance plasticity, that we may really be able to make some dramatic, lasting change.
Okay.